Hello, and welcome back to lesson 7 of the unit 1 of the higher human biology course. Topic 7 is all about cellular respiration. So, we're going to be building upon the knowledge that you learned at National 5 on respiration and obviously going a bit deeper as we always do at higher. So, cellular respiration is going to be split into three different stages. The first stage that we're going to focus on is glycolysis, and glycolysis takes place in the cytoplasm of the cell. Now, we learned that glucose is broken down into 2-pyruvate and 2P back in National 5. And what we're going to be doing today is we're going to be looking at why that is, what happens in between to enable us to get those 2-pyruvate and those 2P. And we'll look at all the stages involved. So glycolysis, generally speaking, the overall reaction is the breakdown of glucose to pyruvate in the cytoplasm. That's the summary of the process. But as you can see from looking at the diagram, it's a lot more detailed than that. Now, it's a process which actually requires us to put energy in order to get those ATP out at the end. So it's split into two different sections here. We have the energy investment phase, the energy investment phase. And during that energy investment phase, we have to put 2P in. And we do that both here and here in the energy investment phase. Now, the reason that we put those 2P in is to give us the energy to carry out something called phosphorylation of glucose into the intermediate one. So the phosphorylation of glucose requires this ATP here. And the phosphorylation of this intermediate molecule also requires that ATP. Okay, so to allow this metabolic pathway to continue, a couple of these reactions require the ATP for phosphorylation of these molecules. So I'll say that one more time. In order for the metabolic pathway to continue and for glucose to be broken down into pyruvate at the end, a couple of these reactions require ATP for the phosphorylation, first of all of glucose and then of the intermediate. Now, all of this stage here, from here down to here, is known as the energy investment phase because we are putting 2 ATP in to this process. Don't worry too much about the NADH and NAD for now. We'll talk a little bit more about them in the next stage, but just note that this reaction occurs here as well. Okay, but we'll come back to it a bit later on. So at the end of the energy investment phase, we have put 2 ATP in for these two phosphorylation reactions to happen. Then we move on to the energy payoff stage. This is where we start getting the ATP back out. And in total, in the energy payoff stage, we get 4 ATP back out. The energy payoff stage results in what we call a net gain of ATP. Okay, those of you who study business and so on will know all about net profits, etc. It's the equivalent, but for biology. So we end up with more ATP because 4 ATP are being produced here and 2 ATP went in. And that gives us our net gain of 2 ATP that we will remember from that 5 was the yield of ATP in this process. And we can see those two processes here, which produce those 2 and 2, in total 4 ATP. And if we remember as well from National 5 that it's the addition of a phosphate to ADP that builds up the ATP molecule and vice versa. The breakdown of ATP produces ADP and a phosphate. And it's that phosphate actually that's used in the phosphorylation, okay? Basically that phosphate gets added to the glucose and becomes this molecule here. And similarly, that phosphate there gets added. That's what phosphorylation is to add the phosphate, the addition of a phosphate molecule. And then lastly, we're in aerobic conditions. And the rest of this topic is going to assume that we are. That means we have oxygen. If that happens, pyruvate will be further broken down. This pyruvate here is going to be further broken down into another molecule. And that molecule is known as an acetyl group. Now, we'll talk more about that in the next slide. But that acetyl group is going to combine with something called coenzyme A to form something called acetyl coenzyme A. But we'll see that in the next diagram here. So we've got that pyruvate that was produced at the end of glycolysis, and it combines with coenzyme A. These two combine together here, and they produce acetyl-CoA when we have aerobic conditions. Now, that allows us to then move on to this stage here, this circular stage here, which is known as the citric acid cycle. Citric acid cycle. Now, that occurs in an area of the mitochondria known as the matrix. We know that the second part of aerobic respiration from National 5 takes place in the mitochondria. We're now going to be more specific about where and about what happens within the mitochondria. So the citric acid cycle occurs in the matrix of the mitochondria. That is very much well worth memorizing because it does come up in the exam very regularly. And another thing, just to add on that note, you should always say the matrix of the mitochondria, not just the matrix, because there are many matrices within the human body. So the matrix of the mitochondria is where this occurs. So in the citric acid cycle, the acetyl group from acetyl coenzyme A combines with the oxaloacetate here to form a molecule known as citrate, which is where the citric acid gets its name from, the citric acid cycle, because we're eventually going to regenerate continuously that citrate molecule there. Okay, so acetyl-CoA combines with the oxaloacetate to form citrate. We can see that happening here. Acetyl-CoA, oxaloacetate forms the citrate. Now, the rest of the cycle, when I was a student at school, I had to learn all of the intermediate molecules here that citrate was broken down into before it became oxaloacetate again. However, you guys are lucky, you do not need to know that. All you have to say is that a series of enzyme controlled steps takes place. These are the enzyme controlled steps all the way around the rest of the cycle here. You don't necessarily need to know the specific molecules, the intermediate molecules here. Now, eventually, citrate is going to be gradually converted back into oxaloacetate. Okay, going around this cycle here, it's converted back into oxaloacetate. And during that series of enzyme-controlled steps, there will be the generation of a little bit more ATP. We can see that here. There will be the release of carbon dioxide. And again, we can see that here in our citric acid cycle. So, couple of products here from all these series of reactions that are happening. Now, in addition to that, there is a set of enzymes called dehydrogenase enzymes. Now, if we break down that word, dehydrogenase enzymes, we know it's A's, which makes it an enzyme, and it works with hydrogen. It's got a role in removing hydrogen here. Now, what it does is it removes both hydrogen ions and electrons from the molecules in the citric acid cycle, and it then passes them onto a coenzyme called NAD, and when that combines with the hydrogen ions and electrons, it forms another molecule called NADH, and we can see that happening over here. So the dehydrogenase enzymes will be working right in this area here to remove hydrogen and electrons and then combine those with the NAD to form NADH. Now, this reaction with the dehydrogenase enzymes, removing the hydrogen ions and electrons, that occurs in both glycolysis and the citric acid cycle. So we see it here in the citric acid cycle, but we also see it back up here in glycolysis in this region here. So that reaction happens in both steps, in both glycolysis and the citric acid cycle. And I'm just emphasizing that because that has been asked in a past paper question before, and it's well worth remembering that. Students often remember it happening here in the citric acid cycle, but forget that it also happens during glycolysis. And then finally, those hydrogen ions and electrons that are part of the NADH are passed on to the next stage of the process, which is known as the electron transport chain, and we'll get to that in a second. And that's located on the inner mitochondrial membrane. So let's look into that. The electron transport chain and ATP synthesis. So the electron transport chain is a series of carrier proteins, okay? That's these things here: carrier protein, carrier protein, carrier protein, protein. We can see that series of carrier proteins there, 1, 2, 3, 4 in the diagram. Now, they're attached to an area called the inner mitochondrial membrane, or the IMM, as I think of it in my head for short. Now, remember, that's a different part of the mitochondria from where the citric acid cycle takes place. That takes place in the matrix. This stage, the electron transport chain, is in the IM, or the inner mitochondrial membrane. That's this area here. Now, the electrons that were brought by the NADH, they brought along hydrogen ions and electrons. The electrons are passed along the electron transport chain, releasing energy. So those electrons, we can follow their path there. There's one electron, there's another one, and another one, and then eventually they leave. So they'll pass all the way along this electron transport chain, this series of carrier proteins, and they will release energy as they do that. Now, why do they need to release energy? Well, that energy is going to allow hydrogen ions, the same ones that the NAD transported, the hydrogen ions to be pumped across the inner mitochondrial membrane. And you can see the hydrogen ions getting pumped through each of these proteins here. The hydrogen ions get pumped across the membrane. Now, the reason they have to be pumped across the membrane is because if we look at the diagram, the concentration of hydrogen ions on this side of the membrane is much higher than this side of the membrane. So they're traveling by active transport. They're going against the gradient, which is why we need the energy from the electrons to actively pump the hydrogen ions across the inner mitochondrial membrane. So I hope you're following me so far. Finally, because we have this high concentration of hydrogen ions on this side, they are going to be able to flow. Okay, they're going to be able to flow, and it's important you use the correct terminology here. They're going to be able to flow back through this protein here, which is called ATP synthase. Okay, ATP synthase, this membrane protein. And as the name suggests, synthase, it's an enzyme. And as its name suggests, it synthesizes or produces ATP. And we can see that reaction happening there, where ATP is generated from ADP and phosphate. So the flow of hydrogen ions through ATP synthase generates ATP. All right. So I think that's the trickiest part. The last wee part is what happens to all of the things now that the ATP has been produced. Well, the hydrogen ions that are flowing through here, they combine with the oxygen that we needed to be present for aerobic respiration, and the electrons, which are now low energy electrons, and they combine together, all three of those molecules, hydrogen, oxygen, and the electron, and they combine together to form the molecule water. They form water. And that makes sense. Hydrogen and oxygen, H2O, that forms water. But don't forget that the electrons also combine with the oxygen. Hydrogen and the electrons plus water plus oxygen equals the water. And the ATP that's used the ATP that's generated here is used to transfer energy to cellular processes, okay, or cellular processes which require energy. Now, at National 5, we learned about those kind of cellular processes. They were things like muscle contraction, protein synthesis, cell division, and the transmission of nerve impulses. But there's way more than just those. Okay, so all of these processes require ATP, and the ATP generated by the electron transport chain is used to help us carry out those other processes and breathe. So that's a long and complicated little process. So I'll just do a quick recap before we move on. There are 3 steps, 3 main stages. We have glycolysis, which takes place in the cytoplasm, and glycolysis is the breakdown of glucose into pyruvate. We'll go back to that one. We have an energy investment phase and an energy payoff stage, which results in a net gain of ATP. The ATP that we put in is used for the phosphorylation of glucose and the intermediates. And when we have aerobic conditions, the acetyl group will combine with the pyruvate. The pyruvate will be broken down into the acetyl group and combine with the coenzyme A to form acetyl coenzyme A. We then have the citric acid cycle, which occurs in the matrix of the mitochondria. During the citric acid cycle, that acetyl group from the acetyl coenzyme A will combine with the oxaloacetate to form citrate. There'll be a series of enzyme-controlled steps where citrate is gradually converted back to oxaloacetate, and we also get the net products of ATP and the release of some carbon dioxide. In addition to that, dehydrogenase enzymes will be removing hydrogen ions and electrons during both glycolysis and the citric acid cycle, and they'll pass those on to the coenzyme NAD, which forms NADH. The hydrogen ions and electrons from the NADH are then passed on to the electron transport chain, which is located on the inner mitochondrial membrane, and it involves these series of carrier proteins. That the electrons will then be passed along those proteins, and they will release energy as they go. This energy allows the hydrogen ions to then be pumped across the inner mitochondrial membrane, and the flow of those hydrogen ions back through the membrane protein ATP synthase, results in the production of a large quantity of ATP. Finally, all of those things that are left over, the hydrogen ions, the electrons, and the oxygen that we've breathed in for the aerobic conditions, they will all combine together to form water. And the ATP, the large number of ATP produced by the electron transport chain, can then be used for cellular processes like muscle contraction, cell division, protein synthesis, transmission of nerve impulses, and many, many others. Folks, so that was quick. We went through quite a lot there. There's a lot to study. I would suggest with this topic, you're going to have to sit and this is one that would be good to start drawing some diagrams, sketch out the glycolysis, the citric acid cycle, and the electron transport chain. Maybe by having a look at your notes first, then covering those up, having a draw at it, see how much of it you can recall, looking back, drawing it again, and so on. Because there are quite a few diagram questions that come up in the exam relating to these. We also get a lot of questions in the exam which are extended response questions, so the longer answer questions. And again, they can be on one or two of these steps in the cellular respiration process. It's rare for you to get asked about all three, but it can be on one or two of these steps. So it's a good idea to memorize as much of the processes and the reactions that happen throughout cellular respiration in case you do get a question on that. But I'm going to move now on to some past paper questions, so join me in a minute to do that. Hi folks, welcome to the past paper section of the cellular respiration topic. So we're going to do some multiple choice questions, one or two section 2 questions, just to apply all of that knowledge, because we're probably all feeling a bit bamboozled by all of the new terminology and all of these various reactions and stages. Trying to get it all together in our head is tricky, but I often find that when you apply the knowledge and you use a bit of active recall, then that's the best way to consolidate things like that. So this question says, during glycolysis, dehydrogenase enzymes catalyze the what? So what they do is they remove hydrogen ions and electrons, and they do it from the citric acid cycle. So we're going to be looking at B as the answer for this one. Okay, B as our answer here. The next one. The diagram below represents some of the processes which occur at the inner membrane of a mitochondrion. Okay, so the inner mitochondrial membrane, IMM. Which letter represents the transfer of the high energy electrons? Now we know that the high energy electrons, they pass along the series of carrier proteins that span the electron transport chain. So which one is going to be that? Well, it looks to me like it's going to be X. That's the one that's traveling all the way through the proteins. Okay, so I think X is going to be our answer here, which is going to be B. Okay, U is the hydrogen ions being pumped through the membrane. I is the flow of hydrogen ions back through the membrane. And Z is the production of ATP through the ATP synthase molecule there. Okay, here's a good one. Which line in the table below describes correctly the function and location of the dehydrogenase enzymes in respiration? So the substance that's removed from the substrate are electrons and hydrogen ions, so it's going to be A or B. And the location is the citric acid cycle. So our answer is going to be A there. Okay, let's see if we can get a section 2 question just for a bit of variety. Let's see. Okay, this one looks good. So the diagram below shows 2 stages of respiration. We've got stage A there, the glucose has been broken down to the pyruvate, and we've got stage B where we've got citrate. So we've to identify stages A and B. So hopefully that's fairly straightforward. We know that glucose is broken down to pyruvate during glycolysis. Okay, if a text box a bit bigger, there we go. And that during stage B here, we have a molecule of citrate. So this will be the citric acid cycle. Okay, citric acid cycle for stage B there. Hopefully those two aren't too bad in terms of starter questions there. Okay, the next question, name substance X. Let's have a look. So substance X here is in the citric acid cycle, and it's the substance which the citrate is eventually regenerated back into. And this substance then combines with the acetyl coenzyme A to form citrate. So that's going to be our oxaloacetate. Part B, during stage A, glucose is converted to pyruvate. Name the molecule that provides phosphate for this conversion. Okay, so what molecule provides the phosphate? Now, we know that a certain molecule, when it's broken down, releases an individual phosphate, and that is ATP. Okay, so ATP provides the phosphate for that conversion. Okay, C, the conversion of citrate to substance X in stage B involves several reactions. Name two molecules apart from the NADH which are produced during these reactions. Okay, so um, again, we're going to have ATP. That's odd for it to have two answers there in the same question. ATP is one of the molecules which are produced, and carbon dioxide. The carbon dioxide is produced as well during this stage. Folks, so that's enough for this lesson. We've gone through a few questions here. Hopefully that's helping to cement that. You can obviously look through more past papers, especially more recent ones, and find some questions there where you can practice cellular respiration. I'm going to do a lot more of those questions as well when I do a Unit 1 summary. I'll be looking at some of the more recent past paper questions during that. So we'll do a bit more respiration past paper stuff. And I hope that that was useful for you. As always, just leave me some comments to let me know what you found useful and anything else you'd like to find out more about or to be covered. And I'll try my best to do that. Okay, folks, take care for now. Class dismissed.